Hey students, it's Mr. Lucido. Just wanted to create a quick recording of the lesson from this morning. So hopefully if you missed anything, you're able to go back and find the part that you need. Again, Unit 5, Lesson 2 was mainly intended to go over simple Mendelian genetics, which is centered around an experiment that likely you are familiar with, having to do with these pea plants. Now, I started this lesson by talking about how prior to Mendel's experiments, there were lots of working theories for how people understood inheritance, how traits were passed from generation to generation, but probably the most common was something called blending theory. And the main idea of this theory was understandable at the time. It was based on the observation that if you looked at, say, a human child, many people thought that the child contained the intermediate of the traits when comparing mom and dad. In other words, if mom was tall and dad was short, that when this child would grow up to an adult, we would expect that child would be having a height somewhere in between mom and dad. And people thought this was true for all traits, that it was the offspring was simply the average of mom and dad's traits, or blended together. And while that could explain some things, it lacked in its ability to explain a lot of other observations people made about inheritance. I brought up the example of sometimes, when we look at our siblings, it seems that maybe one sibling looks more like one parent and another sibling looks more like another parent. And so Mendel, part of his experiment was actually to understand if blending theory was actually how traits were passed along. And so he set out to just conduct this pretty straightforward experiment with pea plants. Now, pea plants produce peas, but they also have flowers. And his most well-known experiment started with taking what he called true breeding parents. He had a series of plants in his garden that were true breeding purple flower pea plants, meaning that generation after generation, all of those plants always produced purple flowers. And he crossed them with plants from another part of his garden that were true breeding white flower pea plants. Same idea, but generation after generation always produced white flower pea plants. And through a pretty well-known process at the time, gardening technique of crossing these plants, he was interested to see what would occur if he crossed these true breeding purple and white flowers. Because blending theory would say that the offspring between these plants should be somewhere in between those colors, like a pale purple or like a light purple. But in fact, what he actually saw in the F1 generation was not an intermediate color, but that all of the plants that came from that cross actually had purple flowers, just like the true breeding purple flower plant. And there were no white flower plants to be found in the F1 generation. And so this at first was very convincing evidence that blending theory, at least when it came to flower color, was not what was going on. And so to further understand this, what he ended up doing was crossing these F1 generation plants, either with themselves or with other F1 generation pea plants, to get the F2 generation. And here is where he started to notice something even more interesting. He noticed that while the majority of the pea plants in the F2 generation were purple, there were a good amount of white flowered plants appearing again that were completely absent from the F1 generation. And so looking at this, he proposed, instead of a blending theory, he proposed the idea that there are heritable factors, which now we know are genes, that can have multiple forms. So he said there were heritable factors for flower color, and you could have the heritable factor that leads to purple color, or a heritable factor that has white as the color of the flower. And he also thought about this in terms of there were dominant heritable factors and recessive heritable factors. And using this framework, he started to try to explain what was going on that led to the results in his experiment. And so he said, okay, for the pea generation, we know that each of these flowers are going to contribute one heritable factor to the F1 generation plant. And it seems like the heritable factor for purple color is dominant to the heritable factor for white color. And so he represented this with letters. He said, okay, let's use the letter P to represent the dominant heritable factor and lowercase p to represent the recessive heritable factor. And so if the purple flower true breeding pea plant contributed the dominant heritable factor for purple color, and the white flower pea plant contributed the recessive heritable factor that's associated with the white color, then that must mean that these F1 generation pea plants each had one dominant and one recessive heritable factor. But the idea of a dominant heritable factor was that you would only see the dominant heritable factor as the phenotype. And so he referred to the combination of factors, or what we call alleles now, as the genotype. But the actual trait that's observed, in this case color, was the phenotype. And so the dominant heritable factor was described as being the factor that shows as the phenotype, even if there is a recessive heritable factor present as well. Which means that the only way you would see the recessive heritable factor show up as a phenotype is if no dominant factor was present. And that helped explain what happened in the next generation. Because you see, if we have all of these F1 generation plants are heterozygous, meaning they have a dominant and a recessive heritable factor, that means if we do a self or a cross-pollination, what we're essentially doing is we're taking two heterozygous genotypes and we are passing down one heritable factor from each of these to the next generation. And so that can either be a dominant or a recessive for both halves of this cross. And the plants that receive a recessive heritable factor from both sides of that cross, those will be our white flowered pea plants, because again, we can only see that recessive phenotype if we have two copies of that recessive heritable factor. These purple flower plants, they can either be what we call homozygous dominant, or uppercase P, or like we saw in the F1 generation, they can be heterozygous. And this, right, they have different genotypes, but they have the same phenotype because, again, that dominant heritable factor or dominant allele is what shows up as the phenotype. And so this helped explain why there was no purple plants in the F1, but then there was in the F2. But then what he also started realizing was that the ratio of the F2 purple to white flower plants tended to be almost always about 3-1, meaning for every 3 purple flower plants, there was 1 white flower plant. And he started to think about this in terms of phenotype ratios that we see right here, right, 3-to-1. But then he also started to think about what would be the genotype ratio. And so to consider that, he started to describe these different types of genotypes or combination of heritable factors as homozygous or heterozygous. Now, homozygous just means you have two of the same heritable factor, and you can either be homozygous dominant or homozygous recessive. And then, as we saw in the F1, heterozygous genotypes mean that the genotype has one dominant and one recessive heritable factor. And he predicted that this genotype ratio, specifically the F2, should probably follow a very similar pattern of, in this case, 1 to 2 to 1. And I'll talk about that in just a moment. But what Mendel discovered was that there were these dominant and recessive heritable factors that for every cross, each parent involved in that cross contributes one heritable factor to the next generation. And depending on the combination or the genotype that that plant receives, that will determine their phenotype. And he was also able to determine for not just flower color, but for many other traits of these pea plants, what were the dominant and what were the recessive forms of those traits. And so from these experiments, one of the big things that he took away were actually two laws of heredity that were based on this experiment. There was the law of segregation and the law of independent assortment. So I want to talk about both of those just for a moment here. So the law of segregation, what I want us to think of when we hear that term is this is the idea that, remember, each of those pea plants could only contribute one heritable factor to the next generation. But, right, remember that F1 plant or the F2 plants ultimately ended up with two heritable factors, right? One from one parent and one from the other. And the law of segregation, all that meant was that there is a completely random chance of which heritable factor or allele gets passed down from each parent, right? So in this case, we can use the idea of a coin flip to help us to think of the law of segregation. This is representing the genotype of the male. And what this is showing is that this male appears to be heterozygous for this trait determined by the letters big R and little r. And it's completely random which one of those heritable factors the father will pass down. 50-50 chance that he'll be passed down the dominant factor or the recessive factor. And same thing for the mom, who in this case is also heterozygous. And so it's often thought of in terms of a coin flip because for every trait, we have two heritable factors, and it's completely random which one we're going to pass down to the next generation. And that's why, as we'll see in just a moment, why we're able to make statistical predictions and determine ratios, very predictable ratios, for the offspring is based on this law of segregation. Now, the other law that was based on his work was the law of independent assortment. And the law of independent assortment. And the idea here was that Mendel realized that when thinking about a particular set of traits. So let's take the color of the pea produced by the pea plant and whether or not it's wrinkled or non-wrinkled. What was discovered was that whatever factor is inherited for one particular trait, like pea color, does not influence what factor is inherited for another trait, say if the pea is wrinkled or not wrinkled. And so this was the idea that traits, or heritable factors rather, independently assort into the gametes and are inherited independent of each other, right? Kind of the opposite of this, which is not the case, would mean, let's say you always inherit, or if a pea plant inherited the dominant factor for one trait, that it must always inherit the dominant factor for the other trait, which is not the case, right? It's always random which heritable factor will be inherited for any particular trait. It's not conditional on another trait. Okay. So we got the law of segregation, the law of independent assortment, are kind of two foundational ideas that were based on Mendel's experiments. Okay, so the last thing that I want to talk about that's kind of based on this idea of law of segregation are Punnett squares. So Punnett squares are these helpful tools that we can use to actually predict, based on the parental genotypes, what will be the expected phenotype and genotype ratios. And so you've likely seen one of these before, but let's do an example here. So if I've got a Punnett square, on one side of this square, I'm going to be representing the possible alleles or heritable factors that can be passed down to the next generation. So let's take an example from the F1. So if I'm crossing two heterozygous pea plants from the F1 generation, on one side, so I'm representing this cross right here, on one side, parent number 1 can either contribute an uppercase P, a dominant factor, or a lowercase p, a recessive factor. Okay, and then parent number 2 can contribute either a dominant or a recessive genetic factor. And then everything that's inside of this Punnett square, we can just distribute to determine all the possible combinations and their probabilities that go along with them. And so, for example, this top left will be a homozygous dominant offspring. This bottom right will be a homozygous recessive or white-flowered pea plant. And then these top right and bottom left will both be heterozygous pea plants, right? Now, if you take a look at the different phenotypes, right, we know that these offspring that have those genotypes will be purple flowered, and only these will be white flowered. And this helped explain not only the phenotype, but also gave insight into what the genotype ratios would be. Right here we see our 3-to-1, our 3 purple plants for every 1 white flower plant. But then we also see, when we compare the genotypes, we have what we call 1 to 2 to 1 ratio, right? Just indicating the different types of genotypes we'd expect from that cross. And that's what we see right here. And that's all based on the fact that it's completely random which heritable factor each parent will pass down. Okay, so those are the main things that we talked about this morning. Let me know if you have any questions, and I hope you have a nice rest of your evening.